Welcome to the 7th Annual Art of Science Gallery at Florida Atlantic University, your hometown university. The Art of Science Initiative is a photography and videography contest that celebrates the creative and experimental spirit of scientific inquiry, inviting researchers to visually express the beauty of discovery. These visual works build meaningful connections among scientists, artists, and the community. Collaboratively, the images form a vibrant dialogue between artists and scientists, offering new ways of seeing research through a lens that honors both fields as expressions of human creativity. We're glad you joined us. Now, let's get started. We have a lot to show you this year. Here's how it works. After listening to each image's story, you'll hear signaling time to listen to the next story in the gallery. About a decade ago, Dr. Chelsea Bennis was on a night dive researching nocturnal marine life when she spotted an octopus with arms stretched out like a parachute. "I was in awe of this animal's extraordinary color patterns, an octopus with a teal hue, and its seemingly effortless movements across the reef," said Bennis, a postdoctoral fellow at the Marine Laboratory in the Charles E. Schmidt College of Science. It was a moment that marked the start of a deeper dive into Bennis's dedication to study the animal, one of the ocean's most intelligent and adaptable creatures. She said that observing these animals in their natural environment sparks curiosity and an endless number of scientific questions. Her most recent research shows how octopuses use their 8 arms to interact with the world. In a study analyzing about 4,000 arm movements, Bennis gained international attention for her discovery of subtle behavioral patterns that could shed light on the evolution of motor control in decentralized nervous systems. In addition to her research, Bennis, also known as Octogirl Online, said she tries to educate the world by bringing marine science to life through underwater photographs and videos. One of her photographs, titled Selfie, Cephie, won first place in this year's Art of Science. The image captures a rare underwater encounter where the octopus, also known as cephalopod, reaches for the camera to seemingly take a selfie. From identifying species to inspiring conservation, Bennis uses underwater imagery to tell stories that make science accessible. Her advice to young researchers? Balance data collection with creative communication, adding that images can transform how people perceive your work. Beyond research, Bennis is an active science communicator dedicated to marine conservation. Octopuses, she said, are powerful ambassadors for ocean health. Their presence often reflects the condition of ecosystems like reefs, mangroves, and seagrass beds. Through fieldwork, art, and education, Bennis said she aims to educate the world about the hidden lives of octopuses, one dive, one image, and one story at a time. Researchers often get an intimate view inside fragile sea turtle eggs before they hatch. While this view is essential to their research, it could also be called artistically beautiful, according to Gabby Carvajal, a doctoral student at the Marine Laboratory in the Charles E. Schmidt College of Science. Each glimpse inside such illuminated embryos serves as a step forward in solving a critical puzzle: how to determine a hatchling's sex quickly and humanely, said Carvajal, who earned second place this year for her image called Sea Turtle Beginning. This photograph shows a late-stage sea turtle embryo silhouetted in its egg with its flipper fully formed and its blood vessels dramatically illuminated. The image, both elegant and intimate, highlights a developmental stage few people ever see, Carvajal said. Beyond the art of the scientific process, Carvajal is leading a project to accurately and quickly determine the sex of sea turtle hatchlings without harming them. For a species whose sex is determined by nest temperature rather than chromosomes, climate change is creating an imbalance for sea turtles, she said. As temperatures continue to rise and rise quickly, nests are producing far more females than males as turtles have environmentally determined sex, Carvajal said. Some years on beaches, we don't see a single male in our samples. That's not sustainable long term, as not producing enough male turtles will eventually push populations toward decline, then extinction. The current methods for determining a hatchling's sex are limited. One method requires raising a hatchling for months until it's large enough for laparoscopic sex identification, which is a specialized technique that only a handful of experts worldwide perform. There's no easy, accurate, or field-friendly way to do this, so we're trying to change that, Carvajal said. Research is an essential step from managing sea turtle populations in an increasingly warming world. Nearly 20 miles above your head, where the sky turns black and the air starts to disappear, a balloon explodes, on purpose, as part of a research mission designed to study how systems behave in near-space environments where extreme cold, pressure, and altitude push engineering to its limits. A photograph of that mission, led by Sky Roof, a mechanical engineering student in the College of Engineering and Computer Science, earned third place in the Art of Science. Those frames we captured changed everything. We had launched balloons before, but we'd never actually saw the moment of the burst, said Roof, president of FAU's Aerospace Experimental Association, a group of future engineers with a vision of exploring space and advancing space technologies that conducted the mission. Roof's image, titled Journey to Space, shows a rear view of a balloon bursting in the stratosphere. The cameras used were mounted to analyze the balloon's behavior and how it interacts with high altitudes, but Roof said he was surprised they also revealed the aesthetic power of engineering. Good engineering is an art, he said. There's creativity in solving problems and beauty in seeing how a system behaves at the edge of what's possible. Roof's research is grounded in real mission experience. He has helped lead multiple near-space launches and spent a summer in NASA's University Nano-Satellite program learning systems engineering alongside Air Force and aerospace professionals. Now he's helping train peers to design FAU's next generation of satellite-ready hardware. Photography lets people feel the science, said Roof, adding images matter just as much as the data. When you show the world from 100,000 feet, no borders, just Earth, it inspires curiosity. That's what research and art have in common. They make people stop, look, and wonder. The next 4 images are this year's top winners for faculty members and students in the field and in the lab categories. The universe is an immense, complex place, but according to Dr. Rob Gross, some of its most stunning visual wonders can be discovered right from South Florida backyards. Gross is a university instructor at Charles E. Schmidt College of Science at Florida Atlantic University. He's also a skilled astrophotographer, blending his lifelong passion for the stars with the technical precision of photography. One of his images, called Rosette Nebula, earned top faculty in the field honor in the Art of Science. The image is a result of 1,260 individual pictures taken throughout 10.5 hours. To capture this detail, Gross used a small telescope about the size of a standard camera zoom lens. By stacking hundreds of images, he filtered out the digital noise and light pollution that would otherwise obscure the nebula's delicate structures. The more and more pictures you take, the better it gets, basically, he said. For me, the process is as much about the math and physics behind the light as it is about the visual result. Gross also uses his astrophotography to engage students. While many of his pre-med physics students may initially find the subject daunting, his image of the cosmos piques their interest. When I show them the pictures, I can see them really start paying attention, Gross said. A lot of them like space. They're really interested in how that works. Gross said he hopes his images inspire others to look up and realize that the beauty of the universe is accessible to everyone from everywhere. The universe is sort of an interesting place, Gross said. There's a lot of things that can be discovered in your backyard. At first glance, the glowing strands in this image may appear familiar, like a web of cells stretching through what looks like a developing nervous system. But that initial assumption is exactly what Dr. Laura Fontinas said she hopes viewers will question when they see this award-winning image. They look like neurons, but they're not, said Fontinas, an assistant professor in the Charles E. Schmidt College of Science. These are radioglial cells, and they are the architects of the nervous system. Fontinas's image, called Radiant Architects, reveals glial cells, the heavy lifters and unsung heroes that lay the foundation for neurons generate new neurons, guide them into place, and support them for life, she said. By illuminating the unseen builders of the brain, Radiant Architects reframes how audiences think about neuroscience and about discovery itself, she said. I want everyone to be able to see and enjoy the science, she said. Art gives us the freedom to think about inclusion as well as aesthetics. I hope people, both academics and non-academics, will pause, look closer, and ask, what am I actually seeing? Collaboration is common in humans, but how far does it extend? This is the foundational question that Erin Mencia is investigating by studying 10 bonded pairs of owl monkeys with special attention on this pair named Kai and Winston. Mencia's picture of the two monkeys earned top honor as student in the field in annual Art of Science photography contest. Photography is a way for people to connect with animals they might never work with, said Mencia, a graduate student in the Charles E. Schmidt College of Science. It helps show that these animals have personalities, relationships, and agency. In the image, Kai cautiously waits behind Winston. The pair curiously watch Mencia as he set up a puzzle box designed to test problem-solving skills and their cooperative behavior to land them a couple of sweet treats of berries or raisins. While the study explores the pair working together to solve the task, it also reveals behavioral differences, including how confident and timid individuals respond differently to challenges. Through Art of Science, Mencia said he hopes viewers pause long enough to see more than just a cute photo and leave with a deeper appreciation for the animals behind the research. They're all individuals, Mencia said. You can't generalize their personalities. That's something people don't always realize. Alejandra Coronel Zagara studies the intricate patterns of coral under a microscope to better understand how disease impacts their growth and integrity. This research is vital to coral restoration efforts as well as protection for the marine life whose lives depend on the coral systems for a habitat, said Coronel Zagara, a doctoral student at FAU's Charles E. Schmidt College of Science. In her coral comparative studies, she has investigated the effects of stony coral tissue loss disease on the coral skeleton. Understanding these changes has implications for coral restoration, where preserving or rebuilding coral structures is key to long-term reef survival. These structural changes matter because the coral skeleton is the foundation of the entire reef ecosystem, Coronel Zagara said. If it's compromised, everything that depends on it is affected. Her winning image, titled Skeletal Secrets, highlights the power of visual research to communicate beyond the lab, Coronel Zagara said. With the naked eye, coral doesn't look like this, she said. Images like this allow people to see what scientists see and to appreciate the complexity that lies beneath the surface. Before a drug can treat pain, addiction, or neurological disease, it must first prove itself at the cellular level, according to Alexandra Nedovic, a doctoral student studying chemistry at Charles E. Schmidt College of Science. That early testing phase, where potential treatments either show promise or fail outright, is where Nedovic said she spends most of her time. Nedovic studies how newly designed drug compounds interact with specific receptors in the brain. Her work focuses on drugs made up of short chains of amino acids that could one day offer safer, more targeted treatments for conditions such as chronic pain and neurodegenerative disease. These cells are the starting point for everything, Nedovic said. If a compound doesn't work here, it won't move forward. Nedovic's image of those living cells, titled Developing New Drugs, earned People's Choice in the Art of Science. The image reveals densely packed cells used in pharmaceutical research to test how drugs behave long before they reach animal models or clinical trials. Nedovic said she took the picture while she was adjusting growth conditions to improve data quality, but when she stepped back, she saw something more. I realized it showed the quality of what early drug discovery looks like, she said. It's complex, it's crowded, and it's where progress really begins. Did you know Florida Atlantic University is ranked as one of America's top 100 public universities, according to the U.S. News and World Report? With more than 170 degree programs, from modernizing healthcare and harnessing ocean energy To unlocking the mysteries of the human brain, FAU offers an incredible educational journey rooted in the belief that creativity and dedication fuel academic excellence. Next up, follow the blue line from the right to hear about this year's Research Excellence Awards. Dr. Anne L. Van Zelst, an assistant professor in the College of Education, earned a top award in the Art of Science for her artwork called Sonic Portrait, which traces the acquisition of the Danish vowel y as in hike. Through repeated attempts, the speaker's pronunciation shifts, each spectrogram capturing a moment of articulation in transition. These 4 images are voice prints, acoustic and visual records of a phoneme being learned. Subtle changes in form and structure reflect the fine motor adjustments of speech. The data were generated and analyzed using PROT, an open-source tool for phonetic research. Together, the images form a sonic portrait of a vowel, a study in adaptation where speech and language take shape through repetition and time. Sharon Hart, professor in the Dorothy F. Schmidt College of Arts and Letters, earned a top honor in the Art of Science for her image called Man in the Ground Vine, which is state-listed as endangered, she said. Hart is researching flora and fauna native to Florida, with an emphasis on at-risk and endangered species. The subjects of the portraits are photographed within their natural habitats or at local conservation organizations. They range from a rare deep red morning glory found primarily in the pine rocklands of Miami-Dade County to the loggerhead sea turtle, Carida carida, which faces multiple threats, including habitat loss, light pollution, and incidental capture by fisheries. The project merges artistic observation with ecological inquiry to reflect Florida's biodiversity. Photography by Dr. Yash Bhagwanji, Associate Professor, College of Education, shows the distinctive hatchet face of a lime tree frog. The picture, which earned a top award in the Art of Science, was photographed near the bank of the only known boiling river in the world called Mayan Tuyacu, in the remote area of the central Peruvian Amazon. Ahmed El-Tawila, a doctoral student at the College of Engineering and Computer Science, earned a top award in the Art of Science for the image called Where Bone Meets Gold. The image depicts false-colored SEM image of 3D printed beta-tricalcium phosphate, beta-TCP, for bone regeneration. Bone has an incredible but limited ability to repair itself, El-Tawila said. In cases of severe injury, infection, or tumor removal, large bone defects require assistance to regenerate. Traditionally, the gold standard treatment is bone transplantation from another healthy site in the same patient. However, this involves a second surgery, increasing the risk of complications and treatment costs. This image shows a synthetic alternative, 3D printed beta-TCP, a ceramic material with a calcium phosphate composition like natural bone. The structure has a multi-scale interconnected porous architecture and fine supporting struts. The manufacturing of such complex geometry is only achievable through 3D printing, resulting in an implant that is both beautiful and functional. The scaffold supports nutrient flow, oxygen diffusion, new blood vessel formation, and provide mechanical support to the neighboring bone, creating a microenvironment where new bone can grow. Over time, the synthetic material biodegrades and is naturally replaced by the patient's own bone. Photographed by Emily Arbsfeld, an undergraduate student in the Harriet L. Wilkes Honors College, this image of fruit fly guts earned a top award in the Art of Science. Titled Face of Digestion, this is a double-labeled fruit fly gut. Animals were genetically modified to add a fluorescent tag, shown here in blue, to a protein, which has been linked to metabolic disorders such as diabetes and obesity. This allows for visualization and study expression differences of the protein in response to different diets, such as high sugar, high protein, or no food. The specimen was also labeled with antibodies against a structural gut protein, shown here in magenta, to visualize the gut structure. Confocal microscopy was used to acquire images of each fluorescent label. To create the composition, different colors were assigned to the labels of the two proteins and displayed as a merged image. Then 2 inverted versions of the picture were assembled to make it look like Escher art. Gladys Sentilme, a graduate student in the Graduate College, earned a top award in the art of science. The image, called Metallic Bloom, was taken using specialized imaging called high-angle annular dark field with a specific type of microscope called scanning transmission electromicroscopy. It uses electrons instead of light to reveal structures far smaller than what regular microscopes can detect. The bright cluster shows nickel particles formed by heating chemicals to 170 degrees Celsius in a polyol liquid called ethylene glycol. The polyol acts like a chemical cooking medium, helping control how the nickel particles form and grow. Though the particles didn't grow on the salt crystal, the surface beneath them looks like one, creating the illusion of a metallic bloom. Each particle is merely a billionth of a meter wide, several times smaller than a grain of sand, yet together they form a branching pattern shaped by heat and chemistry. Dr. Maria Mejia, professor in the Charles E. Schmidt College of Medicine, earned a top award in the art of science for her image called Cracks That Mend, of a fallen tree that reveals the story of time, struggle, and survival. Each ring marks a year of growth, while the cracks running through the wood tell the storms weathered and pressures endured. Despite the breaks, the tree remains whole and offers shelter to new plants and moss along its edges. In many ways, this mirrors the human journey through mental health. Life leaves its marks, some visible and some hidden, but resilience is found in how we continue to grow around the fractures. Healing does not erase the cracks, it transforms them into part of our strength and story. Dr. Megan Davis, a research professor at FAU Harbor Branch Oceanographic Institute, earned a top award in the art of science for her image, Conch Sunset. Sunsets are spectacular at West End, the westernmost point on Grand Bahama Island. On this particular evening, the sunset, the waves, and the adult queen conch shell were so picturesque that many photos were taken. This is a wide-angle shot that captures the beauty of the moment. Davis leads the Queen Conch Lab, which has a partnership project with Blue Action Lab on Grand Bahama Island. Mobile labs are used to raise queen conch larvae to juvenile stage for conservation, restoration, and education. The queen conch is important for the culture, nutrition, livelihood, and coastal seas of countless communities in the Bahamas. As of early 2026, FAU is one of only 13 institutions nationwide to hold a unique combination of 3 prestigious Carnegie Foundation designations. Take a look. Another notable distinction for FAU is that more than 70 of its scientists are among the world's top 2%. Check it out. Research Excellence Awards continue. These newly hatched Florida pompano larvae were reared at the FAU Harbor Branch Oceanographic Institute. At just 0 days post-hatch, they have only recently emerged from their eggs and represent the earliest stage of development for this coastal species. Their bodies are nearly transparent, with organ systems and sensory structures still forming. At this stage, the larvae rely entirely on yolk reserves for energy to support rapid cellular growth. This image, taken by Lee Sun Chin, a doctoral student in the Charles E. Schmidt College of Science and at Harbor Branch, earned a top award in the art of science. In the image, the pompano were reared as part of a research project investigating the ontogeny of Florida pompano to better understand the timing of morphological and physiological changes during early development. This knowledge is essential for improving larval rearing practices and establishing successful pompano culture methods. Tessa Dallow, doctoral student in the Charles E. Schmidt College of Science and the FAU Stiles-Nicholson Brain Institute, earned a top award in the art of science for this image titled Origin Story. This image shows how a developing zebrafish embryo, a powerful model for exploring how the nervous system takes shape. The embryo is curled as it has not yet hatched. It expresses zebrabow, a genetic tool that fluorescently labels many cells, enabling researchers to visualize cell development and interactions in vivid detail. Because zebrafish embryos are transparent, they provide a window into the earliest stages of life, when the foundations of movement and sensation first emerge. The image has been falsely colored for artistic effect, highlighting the intricate beauty of developing structures that guide understanding of both healthy and disordered development. The next few gallery walls are filled with judges' honorable mention picks. Hidden Current by Pooja Perumal Raja, a student at FAU High School and the Charles E. Schmidt College of Science, shows a short-fin mako shark olfactory rosette imaged through contrast-enhanced microcomputed tomography. It shows a segmented rosette, shown in blue, enclosed within its capsule, shown in pink. The rosette, located within the snout, is responsible for odorant detection. It is composed of folded plate-like tissue called lamellae, which increases the surface area for detecting odorants in the environment. Across different species, rosette morphology drastically varies, which could influence patterns of water flow and sensory function. The segmentation visible here highlights the structural organization of the rosette and lamellae in the short-fin mako. At the same time, the surrounding capsule provides protection and support. This is an image of MCF7 cells, breast cancer cells engineered to overexpress the epidermal growth factor receptor, or EGFR. Amplification of the EGFR gene causes resistance to hormone therapy in estrogen receptor-positive breast cancer. The cells were stained with an anti-EGFR antibody. The image, titled Crimson Spread, shows localization of EGFR on the membrane and inside the cell. The image was acquired with an in-cell analyzer 6000 automated high-content confocal imager at 20 times magnification and shows how EGFR spreads through the cell architecture, creating an intricate subcellular distribution pattern. The photographer, Ashley Arbsfeld, is an undergraduate student in the Harriet L. Wilkes Honor College. This photograph, called Spaced Out Eggs, shows Drosophila oocytes at different developmental stages nested next to the fruit fly gut. Dissected preparations were labeled with an antibody against the Drosophila attractin homolog, shown in cyan, as well as with DAPI dye to visualize all nuclei, shown in magenta. The mammalian transmembrane protein attractin is a multifunctional protein with roles in oocyte maturation, energy homeostasis, appetite regulation, immune responses, and pigmentation. Its loss of function has been linked to degeneration of the nervous system and testis, type 2 diabetes, and sleep disorders. The overlaid images acquired with confocal microscopy reveal that, like in mammals, the fruit fly attractin homologous protein is also expressed in oocytes and guts. The image was taken by Tiara Stedman, an undergraduate student in the Charles E. Schmidt College of Science. Gut gathering shows a triple-labeled Drosophila melanogaster, commonly known as a fruit fly, stomach, interior parts of the midgut, and renal tubes. The animal was genetically modified to label the endogenous Drosophila distracted protein with a fluorescent label. Loss of function mutations of the homologous gene in mammals is associated with obesity, type 2 diabetes, and neurodegeneration. This research is about understanding the role of this transmembrane protein in the communication between gut and brain to regulate feeding behavior and neuronal health. The preparation was also labeled with DAPI dye to visualize all nuclei, as well as with a label to visualize the gut. The image was taken with a confocal microscope, and the different fluorescent labels were overlaid in different color combinations. It was taken by Fernanda Salomayo del Bianco, a student dual-enrolled at FAU High School and the Harriet L. Wilkes Honors College. This transverse cross-section reveals the elegant internal anatomy of a turtle grass root, an unseen world beneath Florida Bay's iconic seagrass meadows. Prominently featured in this microscopy image are large parenchyma, or airspace tissues that form a vital internal oxygen highway, transporting photosynthetically derived oxygen from the leaves to the roots. In the sediments where seagrasses grow, oxygen is a scarce but essential resource. These airspace structures are key to avoiding stress associated with low oxygen and enabling respiration where roots cannot rely on the surrounding environment. The root is stained with toluidine blue, allowing researchers to determine what compounds compose seagrass cell walls. Cross-sections like this offer insight into how anatomy supports function. Seagrasses are vital ecosystem engineers, stabilizing sediments, supporting biodiversity, and sequestering carbon. By unraveling the physiology used to survive in challenging environments, researchers gain a deeper understanding of its resilience, which inform efforts to protect and restore these coastal habitats in the face of accelerating change and human impact. This image, titled Seagrass Root, was taken by Alex Hoey, a graduate student in the Charles E. Schmidt College of Science. The image depicts the insulin-producing cells, IPCs, and diuretic hormone 44, DH44 neurons of the common fruit fly, arranged to resemble the fly's head. Initial images were captured using a 40x objective lens on a confocal microscope. Most of the head structure was comprised of a 13-day-old unfed female fruit fly, while the lower proboscis was an 8 to 9-day-old male fly that was fed on a glucose and sucrose-rich diet. The cells were genetically modified to express two different fluorescent markers, Drosophila distracted protein in pink and Drosophila insulin-like peptides in green. IPCs, pink, are analogous to mammalian pancreatic beta cells, which release insulin or insulin-like hormones to regulate high blood sugar levels. DH44 neurons, green, are analogous to mammalian corticotrophin-releasing hormones, which play a crucial role in detecting dietary amino acids, the building blocks of proteins, and promote food searching and consumption behaviors. Both neurons play essential roles in maintaining metabolic homeostasis, keeping the body's internal environment balanced to support optimal metabolic function. Dysfunction in these cell types can contribute to metabolic disorders such as diabetes and obesity, as well as increase the risk of neurodegenerative diseases. This project aims to uncover the mechanisms that drive endolysosomal trafficking, the process which vesicles, called endosomes, transport cellular material to lysosomes for sorting, recycling, or disposal, with a focus on the distracted protein. The photograph, called Fruit Fly Desire, was taken by Catherine Sankey, a graduate student in the FAU Charles E. Schmidt College of Science. Look closely. Do you see the ants marching along the greenest of green water lettuce? Water lettuce is an aquatic plant found throughout the globe, with populations on every continent except Antarctica. The varieties found in Florida are unique because there are multiple genetic varieties that morphologically are practically identical. However, the fascination lies in the fact that one of these variations are native to Florida while another is invasive. This raises questions about how to approach mitigation efforts, according to photographer Angelina Zitelli, a graduate student in the Charles E. Schmidt College of Science. Zitelli earned an honorable mention in the Art of Science for her image called Lettuce Paradox. This reproduction of a Viking jawbone was printed using biodegradable filament in the anthropology lab at the Harriet L. Wilkes Honors College. 3D printing archaeological reproductions offers hands-on learning that brings history to life. Students and researchers can physically interact with accurate replicas of fragile or rare artifacts, deepening understanding through touch and close examination, something often impossible with originals. These reproductions make ancient objects accessible in classrooms, museums, and public spaces worldwide, promoting inclusivity and engagement. They also preserve the form of artifacts that may be too damaged or delicate for handling. For archaeologists, 3D prints support experimental research and reconstruction without risking the original pieces. Additionally, digital models used in printing allow for easy sharing, collaboration, and even virtual reality integration, making archaeological education more dynamic, immersive, and globally connected. Through technology and prints like this, the past is made tangible. The photography, titled Past Reprinted, was taken by Dr. Jacqueline Fuchs, professor in the Wilkes Honors College. This photograph by Dr. Maciej Stawakowski, assistant professor in the Charles E. Schmidt College of Science, is called Lipid Droplets. Organelles once viewed merely as inert fat deposits are now recognized as metabolically active organelles that regulate lipid storage, signaling, and cellular stress responses. Their formation, turnover, and interactions with other organelles are tightly linked to metabolic balance and cell survival. The laboratory develops synthetic fluorescent acylglycerol analogs to visualize lipid droplet dynamics in living cells. These chemical probes reveal how neutral lipids are synthesized, mobilized, and exchanged across organelles, providing real-time insight into lipid trafficking pathways. Lipid droplets have emerged as key players in the biology of neurodegeneration, where disrupted lipid metabolism and oxidative stress contribute to neuronal dysfunction and disease progression. By illuminating lipid droplet behavior with fluorescent light, this research aims to elucidate how altered lipid homeostasis contributes to conditions such as Alzheimer's and Parkinson's diseases. Shown here is a confocal live cell microscopy image of one of the probes, colored magenta, synthesized by undergraduate researcher Christopher Gomez, and its co-localization with the existing lipid droplet marker BODIPI, colored green. Micro-CT images are taken of Octopus Americanus for ongoing research on comparative morphology between local octopus species. Micro-CT offers a non-invasive method of visualizing internal morphology in 3D. This is a cross-section directly below the head of the octopus. Each of the 8 arms are visible, along with the axial nerve cord in the middle of each arm. Images were taken by Natasha R. Weiler, a graduate student in the Charles E. Schmidt College of Science. Using fruit flies as a research model, Dr. Tanja Gaudenschweig is unraveling the mysterious connection of how a person's gut impacts their mood, their brain, their behavior, and even the onset of neurodegenerative diseases like Alzheimer's. Most people think of the gut as ugly because they associate it with its final product, said Gaudenschweig, a professor in the FAU Charles E. Schmidt College of Science. But when you look closely, it's astonishingly beautiful. Gaudenschweig and several of her students recently earned accolades in the art of science. Our images show the elegance that can be found in science while also reminding us how important the proper communication between gut and brain is to human health, Gaudenschweig said. Miscommunication leads to obesity, diabetes, mood disorders, and neurodegeneration. Gaudenschweig's winning image, titled Owls Having Guts, earned an honorable mention. This image reveals the intricate structure of the fruit fly digestive system. Her students' entries reflect their emerging scientific skills, she said. My students spend many hours learning and then executing tedious dissections of these tiny fruit flies to obtain these beautiful images. Seeing them recognized for that hard work is incredibly rewarding. I'm proud of my honorable mention, but watching my students succeed is even better. Photography by Marianne Charlene Monet, a graduate student in the Charles E. Schmidt College of Science and Stiles Nicholson Brain Institute, shows part of the brain where memories begin their journey, the hippocampus, a region essential for learning and memory. The red glow highlights a special group of neurons known as granule cells located in an area called the dentate gyrus. What makes this image unique is the long red tail stretching from these cells into other hippocampus regions called CA2 and CA3. This vibrant projection reflects how the brain encodes, organizes, and communicates new memories. The green signal, labeled brain cells with NEUN, marks other neurons, including pyramidal cells in CA2-3, which are involved in processing and retrieving social memories. Within these neural circuits, the brain begins to transform experience into memory and forms the foundation for how we recall events, recognize others, and navigate the social world. A few additional points of pride for Florida Atlantic University are top rankings for best colleges in the nation, according to the U.S. News & World Report. As your hometown university, enjoy these honors with us. Art of Science judges also chose winning photographs named Top Postdoc and Top Staff. When Dr. Hussam Al-Sharif, a postdoctoral fellow at the FAU Stiles-Nicholson Brain Institute, noticed a maze of looping lines being carved across a small petri dish, he said he did not initially see art, he saw stress. But that unexpected pattern, which Al-Sharif titled Survival, earned recognition in the art of science. It's also a perfect example, he said, of how a routine research observation can translate into a striking visual image. This image shows the movement of microscopic worms as they respond to simulated starvation. Barely visible to the naked eye, these transparent worms are widely used in biological research because they share many genetic pathways with humans and allow scientists to closely observe behavior and biological change in real time. Hussam uses them to study addiction, specifically the effects of amphetamines on health and behavior. In Survival, the worms crawl across and burrow through a jelly-like substance made from seaweed that serves as both a growth surface and food source in laboratory experiments. As nutrients become scarce, the worms trace winding paths through the gel, clustering and scattering as they search for sustenance. The result is a dense network of etched lines that resembles an abstract drawing. While Survival was not created with art in mind, Al-Sharif said he recognized something striking in his observation. The patterns felt expressive, almost communicative, revealing how organisms respond when resources disappear, he said. Some viewers may see a maze, others a body, an animal, or a symbol entirely their own. That ambiguity is intentional, he said. For Al-Sharif, the piece also underscores an important philosophy he carries into his work. Discovery often happens when researchers allow themselves to notice what they weren't looking for. The patterns that became Survival emerged not just from a planned experiment, but from paying attention. This grid of colorful cross-sections may look like abstract art, but the image is actually a series of MRI scans of ordinary fruits, including kiwis, tomatoes, and mangoes. The image, titled Orchard Architecture, by Dr. Rifat Gabur, director of FAU's MRI Center, won a top award in Florida Atlantic University's annual Art of Science Photography Contest. Using MRI, Gabur scanned the fruits to demonstrate how the instrument reveals the internal structure of objects. The same principle used to distinguish tissues in the human body was used to show the structure of the fruit. In the images, seeds, cores, and soft interiors appear as distinct patterns and textures, highlighting MRI's ability to capture subtle contrasts. People usually think of MRI as a hospital machine, Gabur said, but it's really a powerful tool for seeing inside almost anything. Beyond its visual appeal, Gabur said the image reflects his broader research mission at FAU of expanding MRI techniques to study the brain and neurological disease. Through the art of science, he said he hopes the image sparks curiosity and helps audiences better understand how imaging technology works. Next up is the winning video submission in this year's contest. Science doesn't sit still, and neither does Tyler Saravich's research. As a doctoral student in the Charles E. Schmidt College of Medicine, Saravich earned a top honor in the art of science for his three-dimensional video of Huntington's disease neurons. "I wanted to show that research isn't static," said Saravich, who is studying neuroscience at FAU's Stiles-Nicholson Brain Institute. "Science is dynamic, and sometimes the best way to understand it is to actually see it happening." Saravich's winning video, called Expanded Insight, uses a technique known as expansion microscopy to magnify neuronal structures and reveal molecular interactions that are otherwise invisible under standard imaging. The result is a vivid and immersive video that blends advanced neuroscience with artistic storytelling. To push beyond traditional limits of resolution, Saravich physically expands his samples by embedding them in a hydrogel that swells up to 10 times its original size. This process allows researchers to observe minute cellular activity with remarkable clarity. While the technique was developed to answer a scientific question, Saravich said he quickly realized its artistic power. By combining color, motion, and depth, he said his work invites audiences outside the lab to engage with neuroscience in an intuitive way. As we bring this year's guided gallery tour to a close, please enjoy some of our award-winning Science in Seconds videos. Did you ever wonder what it's like to judge the art of science? Here's your chance to find out. This wall shows off the 6 runners-up in this year's contest. Now it's time for you to cast your vote. Lastly, here are some fun facts about the art of science, including a cool tidbit about many of this year's winners being featured in USA Today. The art of science reflects Florida Atlantic University's outstanding contributions to research and innovation. Thank you for being a part of this year's art.